Hello, everyone, and welcome. My name is Simon Roux from the Joint Genome Institute in Berkeley Lab. And on behalf of the VEGA 2021 organizing committee, I'm very happy to welcome you to this fourth seminar in the VEGA 2021 series. So we have a great seminar today focused around the topic and the everlasting questions of virus taxonomy and classification. First, a few logistics. We'll have two main talks here, which will be separated by some flash talks. We encourage all attendees to submit their questions for the main talk through the Zoom Q&A. We'll try to address as many of these questions as we can live. But if there are any remaining questions, or if you want to discuss and interact with the speakers, we'll also encourage you to join the breakout session, which will be a separate and different Zoom meeting at the very end of this webinar. So with that said, I will hand it over to our seminar chair, Eugene Kuhn, to introduce our first speaker, Mart Krupovic. Eugene?
Thank you very much. Thank you very much for the invitation to chair this exciting seminar. Simon, I would like to share my screen now, if I could.
You should be able to, yes.
And I can do that, yes. Yes, indeed. So welcome, everyone. Today, we are going to talk about organizing the global virosphere, why and how, and we have these two speakers, Mart Krupovic from Institut Pasteur in Paris and Jens Kuhn from National Institute of Allergy and Infectious Diseases at NIH here in Maryland. And I'm both delighted and feel slightly awkward to introduce these speakers because we have, as we shall see, we have been working together very closely on these subjects, but their contributions are the main, so I'm more delighted than feel awkward. So, organizing the global virosphere, why and how? First of all, because we can. Because of the rapidly growing diversity of virus genomes and the advent of direct and objective characterization of diverse viromes that exist on this planet, primarily from metagenomics. Second of all, because nothing in virology, as in the other areas of biology, makes sense except in light of evolution. And now they have the opportunities to establish evolutionary relationships across the virosphere, as you will see from these talks, for the double benefit of the increasing diversity of the genome database and the sensitive, powerful methods for protein sequence and structure comparison. Then we must do that. We simply must do that, because otherwise, all this new diversity is in vain. We cannot progress in characterization of new viruses without a robust classification taxonomic framework rooted in the established evolutionary relationships within the virosphere. And finally, last but not least, because the important pathogenic viruses, such as, for example, SARS-CoV-2, do not exist, cannot evolve in isolation. They evolve from other viruses. And if we want to understand them for the long haul, we must look at these deeper evolutionary relationships. So putting the entire virosphere into order. This is a slide from our forthcoming article with Mart, where we connect the so-called Baltimore clusters of viruses, that is the large-scale, high-level classification based on the genome expression replication strategy with the phylogenetically established realms of viruses. This is pretty solid now, as you will probably see from Mark's talk. And finally, that last point. This is a global phylogenetic tree of RNA viruses that we have come up with a couple of years ago, which includes 5 major branches that now are official phyla of RNA viruses. And even if the branching order within the tree might still be up to refining, the branches themselves are very solid. And you can see that these main groups of human pathogenic viruses, coronaviruses, rhinopolio, hepatitis C, for which the Nobel Prize was awarded last year, rotaviruses, flu, Ebola viruses, they're all scattered. They belong to different major phyla level branches within these three. And they are incidentally related to viruses that are not at all pathogenic and do not even infect animals. These are complicated relationships, but we must understand them if we really want to know what these viruses are about. So I will stop sharing my screen at this point, and it is my great pleasure to introduce now Mart Krupovic, the first main speaker at this seminar. Mart is currently the head of the archaeovirology unit in the Department of Microbiology at Institut Pasteur in Paris, France. He received his PhD about a decade ago with Dennis Bamford in general microbiology from the University of Helsinki in Finland, where he already distinguished himself by gaining very important insights into primarily the evolution of the structural component of viruses together with Dennis. At one of his talks, Mart used a sentence to characterize his work that impressed me strongly. He said that my day job is studying archaeoviruses, but for the rest of the 24 hours in the day, I'm trying to study the entire virosphere. And Mart has made major advances in both his day job and his night job. In his day job, he made striking discoveries, such as the presence of the A-form DNA in the particles of several groups of archaeoviruses. Basically, I would say mini revolutionary discovery. But he also greatly contributed to understanding the virosphere, and as I mentioned before, putting the entire virosphere in the order. And I look forward to the discussion of this all-important subject. Welcome, Mart.
Hello, everyone. Many thanks to the organizers and Simon for the invitation, and Eugene for the kind introduction. It is always a pleasure to be presenting in front of an audience who is fascinated with viruses. And I hope I'm not too far off that many of you, if not most of you, are indeed excited and fascinated with the viral world as much as I am. And it is hard not to be, because viruses are everywhere. They impact everything on our planet. And what excites me the most is the diversity of viruses, right? And viruses present a beautiful zoo of virion morphologies and structures. And here you see some of the representative morphotypes we see in the virosphere. And the questions which I always was excited about, almost from my childhood, is where do viruses come from? And specifically, what are the ultimate origins of viruses? And also how all these different groups of viruses relate to each other, all these different looking viruses. And interest in these subjects I share with Eugene, and pretty much everything what I will be presenting today was done in close collaboration with Eugene over the past 5 years or so. So let's start with the origin of viruses. Okay, so historically there are three main hypotheses have been proposed over the years to explain the origin of viruses. And the first one is the viruses early hypothesis, which suggests that viruses are direct descendants of the primordial replicons, which existed very early in the evolution of life, perhaps at the pre-cellular stages of evolution, when all kinds of replication machineries have been invented and tested, and some of them went to become cellular organisms, whereas others were less successful, or perhaps I should say more successful, and became viruses, which parasitized these early cellular organisms. Now, the second hypothesis, known as the regression hypothesis, views viruses as ultimate degeneration products of cellular organisms, which lost some of the molecular machinery, such as the translation apparatus, and then succumbed to obligate intracellular parasitism of cells. And this particular hypothesis has been boosted more recently by the discovery of giant viruses. And the third hypothesis, known as the escape genes scenario, views viruses as genes which escaped from modern cells, and then these genes gained ability to infect and became parasites of cells. And according to this view, genes escaping from bacteria, archaea, and eukaryotes are giving rise to bacterial, archaeal, and eukaryotic viruses, respectively. Now, these hypotheses, they pose some questions. And the first one of which is the timeline. When did viruses originate? Because they propose very different timelines from the pre-cellular stages of evolution all the way to modern cells. And these scenarios, they are mutually exclusive with for any given group of viruses, but perhaps different groups of viruses could have emerged by different mechanisms. Now, also the question is there on how many different occasions this virogenesis have occurred. And perhaps most importantly, it is still not clear from these hypotheses what is the source of major functional modules which constitute the virus genomes. So how did these viral genomes come together to function as viruses as we know them, right? And what we know about viruses, regardless of their size and complexity, is that for all viruses, they have to contain two functional modules, and that is a must. And one functional module is responsible for genome replication, and the other one is required for virion formation, right? In the simple viruses, such as the single-stranded DNA viruses, these two morphogenetic modules are reduced to just a single gene each. This is a genome of the porcine circovirus, which encodes just two proteins. One for genome replication, the rep protein, and the other one for virion formation, the capsid protein, right? And of course, the more complex viruses, they have additional embellishments to these two core components, which leads to increase in genome size and virion size and so on. But when trying to understand the origins of viruses, I think it's reasonable to focus on the more simpler systems first. So how do we go about understanding the origins? Well, the idea is pretty simple. If we are able to trace the origins of these different functional modules, perhaps we can understand where the viruses themselves come from. Okay, so let's start with the virus replication module. So comparison of viral genomes from across the virosphere revealed the presence of several replication enzymes, which are widespread in otherwise unrelated viruses infecting hosts in all three domains of life. And these types of, the most notable examples of these so-called hallmark replication proteins of viruses include the RNA-dependent RNA polymerase found in all RNA viruses, reverse transcriptase in all reverse transcribing viruses, and protein-primed family bDNA polymerases, and rolling circle replication initiation endonucleases. And what is important is that these proteins, they do not have close homologs or sometimes homologs at all in cellular organisms, which would be involved in cellular genome replication. And interestingly, all four of these proteins are based on the very same old RNA recognition motif. And I say old because this protein or this fold is believed to be one of the most ancient folds in the protein universe, dating all the way back to the RNA world. And these types of motifs are found in different RNA binding proteins. Okay, so all these 4 replication proteins are based on this basic motif. And the fact that they are so widespread in viruses from all domains of life suggests that evolution of these replication proteins has occurred very early on in the evolution of the viral world. And this is consistent with the virus early hypothesis, which I talked about earlier. And at the same time, this excludes the escape genes hypothesis due to the lack of any similarity between these replication proteins found across the virosphere and any cellular proteins involved in cellular genome replication, right? So what about the capsid proteins? We have compared capsid proteins and other major structural proteins from across the virosphere from all families of viruses. And what we found is that in many cases, we can find actually cellular homologs for major structural proteins forming the viral particles. And here you see several examples, and perhaps most notable of these is the relationship between the jelly roll capsid proteins, which are found in roughly 1/3 of all virus families, which we currently know about. And this type of, so this kind of capsid protein is widespread in bacterial, archaeal, and eukaryotic viruses. But what is quite striking is that this fold is even more widespread in cellular organisms, in bacteria, archaea, and eukaryotes, where these proteins having exact same folding, they are functionally diverse. And here you see the carbohydrate binding module, which is responsible for binding to sugars and present in all 3 domains of life. And these examples of close relationship between viral capsid proteins and bona fide cellular proteins, they are accumulating, and perhaps, you know, the most recent one was the finding that one of the 2 major structural proteins of the giant Pandora viruses are in fact related to inactivated bacterial glycosid hydrolases. So this kind of the evidence is constantly accumulating on the link between the capsid proteins and the bona fide cellular proteins, which are functionally diverse. Okay, so combining these 2 lines of observations on the replication proteins and the capsid proteins led us to propose a new chimeric scenario for the origin of viruses. And according to this scenario, viral replication modules have evolved very early on in the evolution of life. At the same time, perhaps at the precellular stages evolution, at the same time when the cellular replication proteins have been invented. And some of these replicons in this pool went on to become cellular genomes, whereas others remained as parasites, similar to contemporary plasmids or transposons. But the true viruses have likely originated already at the cellular stage of evolution by acquiring functionally different genes from the cells to function as the viral capsid proteins. And this type of exaptation of different cellular genes to function as capsids for viruses evidently has occurred throughout the evolution of life, from the very early stages of evolution, as in the case, for instance, of the jelly roll capsid proteins, to the modern cells. And we do have some examples of relatively recent recruitment of cellular genes to function as major structural components of virions. Okay, so now let's look at more closely at particular parts of the virosphere. Okay, now that we have this kind of general framework of how viruses have originated. And currently ICTV classifies the virosphere in more than 160 different virus families, many of which are seen on this slide. So, so to try to look more specifically, let's start with the simple viruses, single-stranded DNA viruses, which, as I mentioned, are good models to understand virus origins due to their simplicity. And these viruses, they are widespread in all domains of life, in bacteria, archaea, especially in eukaryotes, and they are very environmentally prevalent as well. And what is common to all these viruses is that they encode homologous genome replication proteins, and they replicate by rolling circle, which is initiated by a specific virus-encoded endonuclease of the HUH superfamily. And homologous enzymes are also encoded by plasmids, which are prevalent especially in bacteria, but also in archaea and eukaryotes, and also in some transposons, which again found in all three domains of life. So this immediately suggests that there is some kind of intertwined evolutionary connection between viruses, plasmids, and transposons. And now we are starting to understand the evolutionary trajectories between these different types of mobile genetic elements. And on this slide, you see kind of a clustering of different families, subgroups of rolling circle replication endonucleases from different families of viruses, plasmids from all three domains of life. And this is clustering based on sequence similarity. And what is obvious is that viruses do not form a monophyletic group in this network, right? Instead, viruses from even the same families, they form distinct clusters with different groups of bacterial or archaeal plasmids, as in the case of these archaeal viruses of the Pleolipoviridae family, or filamentous phages, which are found in 4 different clusters, grouping with different families of bacterial plasmids. Okay, and but perhaps most interesting part of this network is down here, where we see a close evolutionary relationship between one particular group of bacterial plasmids and pretty much almost all eukaryotic single-stranded DNA viruses. And this analysis, it showed that replication modules of the single-stranded DNA viruses are clearly derived from different groups of bacterial and archaeal plasmids. Now, what about the capsid proteins? Here is the tree of the eukaryotic single-stranded DNA viruses, the tree based on the rep protein. And if we now look at the capsid proteins, which are found in these viruses, we see that the capsid proteins are actually polyphyletic, and they belong to different subgroups of the single jelly roll fold. And what is striking is that these capsid proteins, they display specific affinities to different families of eukaryotic RNA viruses, right? And this kind of combining the understanding of the replication protein origins and the transfer of the capsid proteins from RNA viruses brings us to a kind of a model of how we think of the evolution of or origin of the single-stranded DNA viruses in general. So in all likelihood, the diversity of single-stranded DNA viruses has evolved from a pool of rolling circle replication, replicating bacterial plasmids upon acquisition of different capsid protein genes from bacteria to give filamentous phages, archaea, pleomorphic viruses or icosahedral viruses in bacteria. And all the diversity of single-stranded DNA viruses in eukaryotes has been apparently seeded on at least 2 occasions from related plasmids upon acquisition of capsid proteins from pre-existing RNA viruses. Another interesting observation which we made in this study is that small double-stranded DNA viruses of the polyoma and papillomaviridae families apparently have also evolved from the midst of the diversity of single-stranded DNA viruses. And this is also clear in the bipartite network analysis, which you see here, where polyoma and papilloma viruses form a separate supermodule, which is barely connected to the rest of the double-stranded DNA virosphere. So double-stranded DNA viruses in this global network of double-stranded DNA viruses, apart from polyoma and papilloma viruses, we can recognize 3 other supermodules, which all include many different families of double-stranded DNA viruses. And the 2 biggest ones are represented by, again, large number of families, virus families, which in fact hosts in all 3 domains of life. But what is specific to each supermodule is a distinct morphogenetic module, or kind of set of proteins required for virion formation, for genome packaging, virion maturation, and assembly. So the first of these supermodules, which we can call HK97 major capsid protein supermodule, includes all tailed bacteriophages and related archaeal viruses, as well as the eukaryotic herpes viruses. And they have the particular major capsid protein and particular genome packaging machinery, and also share how the particles maturate and are assembled. And the second supermodule corresponds to viruses with a very different major capsid protein known as the double jelly roll major capsid protein. And again, this supermodule includes multiple families, all the giant viruses, mimiviruses and such, but also smaller adenoviruses, bacterial tectiviruses, and archaeal pluriviruses. Right. So where do these capsid proteins come from? So again, I will repeat myself pretty much here, is that the double jelly roll capsid protein, we can trace back the origin of this capsid protein to cellular proteins. So it is believed that the double jelly roll capsid protein, which consists of 2 single jelly roll domains, has evolved from 2 separate proteins, and we indeed have such proteins and such viruses with 2 major capsid proteins, each of which have a single jelly roll fold. And these types of viruses, they are found in bacteria and in archaea. And then we can make a step back to double-stranded DNA viruses where capsids are made from a single major capsid protein with a single jelly roll fold. And then we make a leap from this single jelly roll major capsid protein to cellular single jelly roll proteins. Now, in the case of the second supermodule, the Hong Kong 97 supermodule, which includes all tailed phages, the story is pretty much the same again, although the source of the capsid proteins is different. So in this case, the major capsid protein appears to be a composite of 2 different domains, the DUF1884 and the dodicin-like protein, into which this DUF has been inserted. And you can clearly see the similarity between these 2 domains and the 2 domains in the major capsid protein found in tailed phages. Okay, now the fourth supermodule, and this supermodule is very poorly connected to the rest of the double-stranded DNA virosphere, and it mainly consists of hyperthermophilic archaeal viruses. And this is, as Eugene mentioned, is my day job, so they are particularly dear to me. And if we zoom in on this supermodule, what we find is that it consists of multiple families of archaeal viruses, and these families are connected, but they are not connected through the shared major capsid proteins or through the replication proteins. Instead, they are connected through proteins involved in virus-host interactions, such as the transcription factors, which is most widely shared between these different families, or glycosyl transferases. And these types of proteins involved in virus-host interactions, they are known to be horizontally transferred between viruses or independently acquired from the hosts on independent occasions. So what this means is that this archaeal virosphere consists of multiple families which are not related to each other and apparently not connected to the rest of the virosphere. Okay, so here we have potentially many independent cases of virogenesis. Now, we can kind of map all this diversity of different groups of viruses onto bacterial and archaeal phylogenies, and then try to infer what kind of viruses were associated with the last bacterial common ancestor, archaeal common ancestor, and all the way down to the last universal cellular ancestor. And this type of mapping suggests that the last universal cellular ancestor, Luca, has been already infected by a very diverse virome consisting of diversified viruses with the Hong Kong 97 proteins, similar perhaps to the contemporary tailed phages, but also by tailless viruses with single and double jelly roll capsid proteins. Then the last bacterial common ancestor apparently was already infected by filamentous phages, as well as 5X174-like microviruses. But we cannot exclude the possibility that these two groups of viruses were already present at the stage of the Luca, but then lost in archaea. And conversely, last archaeal common ancestor apparently has been infected by lemon-shaped and rod-shaped viruses, but they could all well be also in Luca, but lost in the bacterial branch of the tree of life. But many families of these archaeal viruses apparently have evolved within particular lineages of archaea because based on our current knowledge, their distribution is very narrow, but perhaps this will change with more sampling. Now, just a few words about RNA viruses. Okay, so RNA viruses in a way resemble single-stranded DNA viruses, and specifically in that they all share the replication module, and namely the RNA-dependent RNA polymerase, which can be used as a marker for all RNA viruses, pretty much like 16S ribosomal RNA genes are used for cellular organisms. And now this allows us to put them all together into one framework. But if you look now at the morphogenetic module of RNA viruses, what you find is that they are extremely diverse, having all kinds of virion morphologies and structures. And even among the viruses which share relatively closely related RNA-dependent RNA polymerases, there can be viruses with icosahedral, rod-shaped, filamentous, enveloped, all kinds of different virions, which indicates that there has been extensive exchange of the capsid protein genes in this part of the virosphere from other viruses, but also from cellular proteins, like in the case of alpha viruses here. Okay, now returning to these 160 families of viruses, different structural, biochemical, functional, and comparative genomic studies revealed kind of common themes among these different families of viruses, which allow us to better understand the organization of the virosphere into evolutionarily cohesive assemblages. And now we can see more clearly kind of the part of the virosphere consisting of these Hong Kong 97 capsid encoding viruses, double jelly roll viruses, single-stranded ones sharing the rolling circle replication mechanism and the underlying protein, and the RNA viruses. And all this allows us now to formally kind of recognize all these evolutionary relationships and embed them into the taxonomy. And this is where I have to give floor to Jens. But before I do so, I would like to thank many people who were involved in different aspects of the work which I just presented, and primarily Eugene and his group at NCBI, but also Valerian Dolia, Arvind Varsani, Darius Kazlauskas, and Jens. And thank you very much for your attention.
Thank you very much, Mart, for what I at least consider to be an exciting talk. Let us check the questions. We have, no open questions. No answered questions, no opened questions. So I guess everything is excessively clear.
No. In such cases, I would like to think that I left the audience speechless.
Well, this being the, yes, you left the audience speechless, stunned. But perhaps not me so much, because I'm familiar with much of the material. So let me ask you a question. Now I will just speak. I will not use this facility using my privilege as a chair. I will just speak. So what do you think of the future? In the future of the exploration of the virosphere, and I have two questions that, I have three questions that bother me all the time, and these are the following: with the further exploration of the diversity of the virosphere, primarily through metagenomics, but through other kinds of genomics as well, are we going to arrive to a significantly greater level of certainty about the ancient stages, about the virome of Luca and last Leca, last eukaryotic common ancestor, etc., etc. You presented what I think is robust inference, but are we going to get qualitatively better in understanding these questions? That's one of my burning questions. The second one is this. You have given us some outlines of the megataxonomy. Jens will give more. But what do you think about how many new realms, let us say? How much more expensive with this megataxonomy is going to become? And thirdly, but not least, the dark matter of viral genomes, so many unknown genes. Are we going to get some straightforward answers regarding to what these are and what they do?
Okay, yes, thanks, Eugene. So the first question, which was?
Which was a simple one. With the further exploration of the diversity of the viromes, are we going to really nail the ancestors? Okay.
Yes, I think we will, and because I'm most certain that we will, because we still do not know so much about, let's say, archaeal viruses. Right, we have explored only viruses in two branches of the archaeal tree, and the rest is very much in the dark, which should help us to understand what was the virosphere of the last archaeal common ancestor and the Luca as well. Right, and in bacteria, I think we are also far from having explored many of the bacterial branches in terms of viruses. So this will fill the gaps and expand the virosphere of Luca, I would expect.
Oh, okay, thank you. That is up to me. That is promising. And I look forward to that. And the next question, was how far is the taxonomy expanding?
Yeah. So
Any new major realms of viruses, anyone, so to speak.
Major realms, probably.
Yeah. I'm putting the plank high.
Yes. Well, major realms probably not, because the four major realms which we have cover pretty much most of the biosphere. So there are some families of viruses which are still kind of were gray on my slide, not belonging to any realm, but these are just a few families which are not very large, right? So we might have, say, 5 up to, well, very optimistically 10 new realms, but I think that's about it. And it's not that we are discovering new, completely different groups of viruses very often anymore. So all these metagenomic studies, if the study which we worked together on, where you kind of double the diversity of viruses and you don't find any new phylum level virus even there, right? And for DNA viruses also, we keep on expanding the diversity of giant viruses, but they all seem to be falling either within existing groups or between these groups, but still clearly fitting in those realms.
Okay, thank you very much. And still, if you will, a little more optimistic or maybe more agnostic, whatever. I agree that we are not going to discover new major ones, but small ones, perhaps couple thousand at the end of the day. All right. And my final question was about the dark matter, but I will sort of merge it. The question from Jeff Blanchard, from the audience who says, well, I will read it. Hi, Mart, wonderful talk. How many of the giant virus structural proteins do you think come from eukaryotes, or are some from bacteria? So maybe you address these and maybe say a few words on the dark matter of the viral genomes in general.
So is the question specifically about the capsid proteins or structural proteins?
I do not know. It says structural. My uneducated guess is that Jeff may not have necessarily meant structure, more generally.
Well, I will say about the structural proteins, starting with structural proteins. Okay, so the major capsid protein of giant viruses is very clearly a double jelly roll protein, which can be traced to much smaller viruses, which infect bacteria, archaea, and so on, and at the pre-LUCA stages. The minor capsid protein can be traced to these well-known small viruses. They encode all kinds of minor structural components for which the functions are generally not known for any of the big viruses. And the origins, as in the case of, for example, this Pandora virus protein, which I briefly mentioned. So this protein is also a minor component of the giant viruses such as mimivirus, but then it switched the function to become major structural protein in Pandora viruses, but their origin is clearly from bacteria. And so I wouldn't expect there to be much innovations of the major structural proteins in giant eukaryotic viruses, but some minor components could have been repurposed and adapted to serve some roles in these. As going for the rest of the genes, it's hard to say, right? Especially for, I think it is well known and from your work and work of others, what is the fraction of bacterial genes and what is the fraction of eukaryotic genes in giant virus genomes. And of course, there are many genes without known source, provenance. And those are the dark matter which you mentioned. And I think we can learn about them in the future when we still improve our methods for structure prediction and kind of deep evolutionary kind of or more sensitive methods for finding the sequence similarity based on perhaps at the fold level, like AlphaFold 2, when it becomes available. I think it will open our eyes on this dark matter a bit more.
Okay, thanks very much, Mart. We do not have much time here. So I will ask the last question with regress from Paul Rivares. Question that may be deviated from the topic, but how can we consolidate viroid evolution in the bigger picture of the virosphere? Is there any direction or initiative for that? And then okay, and I think the rest of the question will come in the breakout room.
Okay, so viroids, I think they are not part of the virosphere or viruses as we currently define viruses, right? So this is they have some features in terms of being infectious, which are similar to viruses, but I would put them in a different category. In terms of classification of viroids, this is something which Jens will probably tell us something about, but they are dealt with by the ICTV, and in pretty much the same way as ICTV deals with viruses, classifying them into different taxonomic groups, except that they are a little bit separate.
All right, thank you very much, Mart. There are more interesting questions, but we cannot really address them now, very hopefully in the breakout rooms afterwards. And now I turn the podium to Simon again to introduce your flash talks.
Thank you.
Thank you, Mart and Eugene. That was fascinating. And yes, for the remaining questions, we can have the breakout session. I mean, we will have the breakout session afterwards, but I know Mart can also try to answer some of them in writing in the Q&A. And in the meantime, let me share my screen so we can move on to, okay, here we go. Some flash talk. And the first flash talk on deck was Sophia.
Hi, can you hear me?
Yes.
Okay.
Go ahead.
A second. So, hi everyone. I'm Sophia from the Charité Institute of Virology in Berlin, and I will present you data on insect virus discovery and the evolution of single-stranded RNA viruses. So, current knowledge is mainly based on viruses infecting humans and animals, but recent studies showed that a much larger virus diversity exists in insects. However, because mostly blood-feeding insects are in focus, we aim to examine virus diversity in the insect organismic group as a whole. For this purpose, we collaborated with the OneKite project, where over 1,000 insect transcriptomes were sequenced. We used profile hidden Markov models built on RDRP alignments for virus sequence detection and inferred phylogenetic trees focusing on current virus taxonomy. For viruses with segmented genomes, we searched for additional segments and inferred co-phylogenetic trees. The Orthomyxoviridae family on the left, known for containing influenza viruses, which you can see at the bottom, showed a much larger insect virus diversity than previously thought, and we see that a clade structure emerges according to the insect hosts. We detected all 3 RDRP segments in 31 cases, and co-phylogeny inferences showed congruence between segments in major clades. Now for Flaviviridae, on the right, insect viruses were not detected in the vertebrate infecting genera, which is expected. We also didn't find new viruses in the genus Flavivirus, so the absence of insect viruses in a traditionally blood-feeding genus suggests that Flavivirus evolved in blood-feeding arthropods. In the clade of Genome viruses, recently discovered in ticks and insects, we found 9 new viruses. Genomes consist of 4 segments and show amino acid similarity in the NS3 and NS5 flaviproteins. Tick-associated segments have a 3' end poly-A tail, conserved 5 and 3' ends, and a class 2 viral fusion protein domain, features that differentiate them from the insect Genome viruses. Finally, the discovery of Genome viruses suggests that segmentation in Flaviviridae evolved probably once because of the monophyletic nature of Genome viruses. Thank you so much. I think that was my time. 2 minutes,
Thank you.
right?
Yeah, that was great. Perfect.
Thanks.
Thanks so much. Next one is Tiani.
Okay, so hello everyone. My name is Kenyi Chang. I'm from Curtis Sutto's lab from UBC. So arthropods are the largest group of animals on Earth, and they have this broad ecological connections with different organisms, and you can find them almost everywhere. But in the past, we mainly focus on those arboviruses that infect human and economic plants and animals, so we actually miss the true picture of their virome. So in this work, we systematically investigate metatranscriptomes from over 700 arthropod species, and we uncovered more than 1,300 previously unknown RNA viruses. And those viruses represent 822 novel evolutionary groups of viruses at a level between the species and genus based on their RDRP proteins. And you can find that those viruses they fill the major evolutionary groups, they fill the major evolutionary gaps in all the major RNA virus groups and across the 5 major branches. And we also found that most of these viruses, they share a significant sequence homology with endogenous viral elements of arthropods. So that means these viruses probably infect the arthropod and also imply for a deep evolutionary relationship between the arthropods and the major RNA virus groups. And what's more is that we also we conducted a co-phylogenetic analysis, and that indicate the RNA viruses of arthropods have commonly co-evolved with their host. And based on this data, we demonstrate that the central role of arthropods have played in the global evolution of RNA viruses. So if you are interested in this work, please find me in the poster session. Thank you.
Thanks. Yeah, RNA viruses are super diverse, but when you start to get into these endogenous viruses, it's mind-blowing. Okay, great. Moving on to Juan.
Hi, everyone. I'm Juan Carlos. I am from the University of the Balearic Island, and today I would like to present the tool VPF class, which is a tool to obtain a taxonomic assignment and host prediction of an input viral sequence. The tool is available at the GitHub, and the classification is provided for both taxonomic and host prediction at different taxonomic levels. The classification is provided for every single viral sequence, and it is not required to download any database to map the viral data. And the user can select the parameters to customize the coverage and accuracy of the input set. This tool is published in the bioinformatics, and this is based on the classification of a set of viral protein families. In the first step of this classification, we try to use annotated databases, which are a set of isolated reference viruses, to classify the VPFs. Then a second step where we classify some uncultivated viral genomes, and in the third step we use some of them to provide a final taxonomic classification and host prediction of the VPFs. And in the right, you can see an example of an output of our tool at the genus level with 3 parameters about membership ratio, virus hit score, and confidence score. And thank you.
Thank you. Very, very useful tool for sure. Next one is Sabrina.
Hi everyone, my name is Sabrina. I'm a current PhD student in Eddie Holmes' lab in Sydney, Australia. And I'm just going to give a quick overview of a huge RNA virus discovery and ecology project I just wrapped up earlier this year. So essentially, I was lucky enough to get to analyze a massive data set from China covering 32 different environments across 17 provinces, including environments like sediments, water, forests, farmland, and animal feces. So we worked out the proportions that different viral families make up in each of the 444 libraries in order to get the quite colorful figure I have here on the left, which describes how virome composition differs depending on the environment sampled. As you can see, some of the general trends are that there is a remarkably large proportion of viruses belonging to the fungi and bacteria-associated Nana-Levy clade in virtually all environments, especially in biofilm and semi-desert soil samples. One more specific observation is that there is a large proportion of Patytypica burner viruses, that's the teal color. They have plant, fungal, and protist hosts, and there's a large proportion of them in animal feces, likely due to the host's diet rather than reflecting any actual infection. Now, if I can take your attention to the red and black figure of phylogenetic trees on the right, this is the key part of the study, which was the discovery of 2,683 novel RNA virus species in virtually all RNA virus groups, shown here as the red tips in these trees. This includes over 1,000 novel Nana-Levy viruses, which really showcases the power of metagenomics in regards to the expanding the known biosphere. So that's me done. Happy to answer any questions in the breakout room later on, or feel free to shoot me an email. Always happy to chat about viruses. Thanks so much.
Thank you. And last but not least, Sana.
Hello, everyone. My name is Sana, and today we heard a lot about the origin of viruses and their evolution. But what's the origin of viruses in the human gut, and how do they develop within the infant virome? To study that, we extracted DNA from total microbial community and virus-like particles from the fecal material of 30 mother-baby pairs who collected their samples longitudinally during pregnancy and 3 months after birth. Although the virome community was different between mothers and babies in terms of viral diversity and composition, still some viruses were shared between the related mothers and babies. Although this number was relatively low and accounted for 11% of all viruses ever detected in a baby. However, we also saw that the bacteriophages dominate the infant gut virome, and more specifically, the temperate bacteriophages, supposedly in their active state. Therefore, we searched through the total microbiomes of mothers that contained bacteria and their prophages for the temperate phages detected in babies. And we indeed saw that the number of shared viruses between related mothers and babies increased dramatically when the bacterial prophages were taken into account. Our results suggest that the first viral colonizers of the infant gut are the temperate bacteriophages, excising from the bacteria that were vertically transmitted from mothers to babies. Thank you for your attention. If you want to know more details about it, I have a poster available, and I will be later available in the breakout room. Thank you.
Fascinating, thank you. And thanks to all Flash Talk presenters. That was awesome and also really on time, so great.